Hello everyone. Today I will introduce an important enabling technology in modern molecular biology, magnetic particle-based nucleic acid extraction. This method has become a standard approach in many workflows, including PCR, sequencing, and molecular diagnostics because of its speed, scalability, and compatibility with automation. At CD Bioparticles, we develop functional magnetic silica materials designed to support efficient nucleic acid purification across a wide range of applications. To begin, let's first understand why rapid nucleic acid extraction is such a critical step in molecular workflows. Nucleic acid extraction is the foundation of nearly every molecular biology workflow. Whether we are performing PCR, quantitative PCR, next-generation sequencing, or pathogen detection, the quality of extracted DNA or RNA directly affects downstream performance. If inhibitors remain in the sample, or if nucleic acids are degraded, the result may be false negatives, reduced sensitivity, or unreliable sequencing outcomes. Because of this, rapid, efficient, and reproducible extraction methods are essential for both research and diagnostic laboratories. Traditionally, nucleic acid extraction relied on several well-established methods, but these approaches have notable limitations. Historically, nucleic acids were extracted using methods such as phenol, chloroform extraction, salt precipitation, silica column purification, or ion exchange chromatography. While these methods can produce high-quality nucleic acids, they are often time-consuming and involve multiple centrifugation steps. Some methods also rely on hazardous organic solvents, which introduce safety concerns and increase handling complexity. In addition, many of these approaches are difficult to automate, making them less suitable for large-scale or high-throughput applications. To overcome these challenges, researchers began developing new technologies, leading to the emergence of magnetic particle-based extraction. Magnetic particle technology represents a major advancement in nucleic acid purification. Instead of relying on centrifugation, magnetic particles allow rapid separation using an external magnetic field. When the magnet is applied, particles carrying bound nucleic acids move quickly to the side of the container, allowing the liquid to be removed easily. This simple physical principle eliminates many manual steps and significantly reduces processing time. As a result, magnetic bead-based extraction has become widely adopted in both research laboratories and clinical diagnostics. To better understand how this technology works, let's examine the structure of magnetic silica particles. A typical magnetic particle used for nucleic acid extraction consists of three functional components. At the center is a magnetic core, usually made of iron oxide materials such as magnetite. Surrounding this core is a silica shell, which protects the magnetic material and provides a chemically stable surface. Finally, the outer surface can be functionalized with specific chemical groups such as hydroxyl, carboxyl, amine, or oligo-DT groups. These surface modifications allow the particles to interact selectively with nucleic acids. Now that we understand the structure, let's explore the mechanism by which nucleic acids bind to these particles. The binding of nucleic acids to magnetic silica particles is primarily driven by electrostatic interactions. DNA molecules contain negatively charged phosphate groups along their backbone. In the presence of salts, cations form bridges between the DNA and the functional groups on the particle surface. This creates a stable interaction that allows nucleic acids to bind efficiently to the particles. After washing away impurities, changing the buffer conditions allows the nucleic acids to be released, producing purified DNA or RNA. Using this mechanism, magnetic beads enable a streamlined workflow for nucleic acid purification. The typical magnetic bead extraction workflow consists of several simple steps. First, the sample is lysed to release nucleic acids from cells or viruses. Next, magnetic particles are added to bind the nucleic acids. A magnet is then applied to separate the particles from the liquid phase. After washing to remove contaminants, purified nucleic acids are finally eluted in a clean buffer. Because centrifugation is not required, this workflow reduces handling time and lowers contamination risk. When compared with traditional methods, the advantages of magnetic bead technology become even more apparent. Magnetic particle-based extraction offers several key advantages over conventional approaches. First, it significantly reduces operation time. Second, the elimination of centrifugation simplifies workflows and reduces mechanical stress on samples. Third, magnetic systems are highly compatible with automated platforms, making them ideal for high-throughput laboratories. In addition, the use of non-toxic reagents improves laboratory safety and environmental compatibility. Overall, these benefits make magnetic bead extraction one of the most efficient purification strategies available today. Despite these advantages, there are still technical challenges that must be carefully managed. One common challenge is balancing speed with extraction yield. Faster protocols may sometimes result in incomplete nucleic acid recovery. Another concern is the presence of inhibitors in complex samples such as blood, soil, or wastewater. Additionally, harsh chemical or thermal conditions can damage nucleic acids and reduce data quality. To address these challenges, optimization of buffer composition, surface chemistry, and workflow design is essential. Advances in particle engineering have led to specialized magnetic beads designed for specific applications. Modern magnetic particles are available with a wide range of functional surfaces to meet different experimental needs. For example, carboxyl-modified particles are commonly used for DNA purification. Oligo-DT functionalized beads are widely used for mRNA isolation. Streptavid-encoded particles enable highly specific biomolecule capture through biotin interactions. These specialized designs allow researchers to tailor extraction workflows for genomic DNA, viral RNA, cell-free DNA, or other targets. At CD Bioparticles, we offer a diverse portfolio of magnetic silica particles designed to support these applications. Our magnetic silica particle platform includes a broad selection of particle sizes and functional chemistries. For example, we provide virus nucleic acid magnetic particles around 150 nanometers in size, which are optimized for viral detection workflows. We also offer carboxyl functionalized particles and larger particle sizes up to several micrometers to support different binding capacities and automation systems. In addition, specialized particles are available for applications such as circulating cell-free DNA and HPV detection. In addition to particles themselves, integrated purification kits are also essential for streamlined workflows. To further simplify nucleic acid isolation, CD Bioparticles provides complete extraction and purification kits designed for specific sample types. These kits support DNA purification from blood, tissue, plants, bacteria, wastewater, and dried blood spots. By combining optimized buffers with high-quality magnetic particles, these kits enable consistent performance across different workflows. Importantly, many of these systems are compatible with automated platforms, making them suitable for large-scale testing environments. With these tools in place, magnetic bead-based extraction now supports a wide range of real-world applications. Magnetic particle-based nucleic acid extraction is now widely used across many industries. In clinical diagnostics, it supports pathogen detection and genetic testing. In research laboratories, it is used in sequencing library preparation and gene expression studies. Environmental scientists use it for monitoring microbial populations in water and soil. It also plays an important role in forensic analysis and food safety testing. To conclude, let's summarize the key advantages of this technology and its role in enabling modern molecular workflows. In summary, magnetic particle-based nucleic acid extraction offers a fast, efficient, and scalable solution for modern molecular biology. It reduces processing time, improves purity, and supports automation across a wide range of sample types. At CD Bioparticles, our magnetic silica particle platforms and nucleic acid purification kits are designed to deliver reliable and reproducible performance for research and diagnostic applications. We are committed to supporting scientists and laboratories by providing high-quality materials that enable faster, cleaner, and more reliable nucleic acid workflows. Thank you for your attention.